Hi, I'm Kristen.
I'm Kelsey.
I'm Laura.
And we're going to be talking to you today about mitosis.
Today let's take a look at all the phases within the cell cycle that are related to mitosis. The cell spends most of its life in the interphase, and it is during the interphase that the cell undergoes preparations for mitosis, which when finished will produce two identical daughter cells from this single parent cell. So here is our parent cell, and within the parent cell, we also need to include the genetic material that will be duplicated and then passed onto the daughter cells. The genetic material is called chromatin, and when found in long strands is also known as chromosomes. These chromosomes are housed within the nucleus, which is surrounded by a membrane known as the nuclear envelope. So here I'm going to draw the chromosomes that are about to go through the replication process to form sister chromatids. In addition to the chromosomes, there is also a structure in the nucleus known as the nucleolus, which is only present during the interphase and is the site of ribosome synthesis and assembly. And then outside the nuclear envelope, within the cytoplasm, is a pair of centrioles, which are involved in the organization of the mitotic spindle fibers in the completion of a cytokinesis or splitting of the cytoplasm, which we'll take a look at later. From the interphase, the cell enters the process of mitosis, which includes four phases: prophase, metaphase, telophase, and anaphase, followed by a cytokinesis. The first phase of mitosis is called the prophase. At this point, several changes have already occurred in the parent cell, and some changes have just started to take place. The nucleolus has already disappeared from the nucleus, and the nuclear envelope is now just also beginning to disappear. The chromosomes in the nucleus have replicated and are now becoming more coiled and visible. Each duplicated chromosome is known as a pair of sister chromatids, and the sister chromatids are connected at their center by centromeres. A full set of sister chromatids was created during the interphase when all chromosomes in the cell were replicated. So here are my sister chromatids in the very recognizable X shape we're so used to seeing. In the cytoplasm, the mitotic spindle fibers consisting of microtubules and other proteins form between the two pairs of centrioles. So here are my centrioles. And then in between these centrioles, I have the spindle fibers start to form. And then these two pairs of centrioles begin to migrate to the opposite poles of the cell. So here are my spindle fibers, and that's prophase.
So now I'm showing you metaphase, which is the phase right after prophase. And here I am outlining the cell for you. And next I'm going to draw in the centrioles, and there will be two at each pole. So I guess you could say two at the North Pole and two at the South Pole. And I will label them for you. Next, I'm going to draw in the spindle fibers. And these are really important because these are going to be what pulls the chromatid to the different poles. So right now, and what I'm about to draw, will be the original chromosomes of the first cell and their duplicates. So I labeled the spindle fibers for you. But I'm going to draw, here's one chromosome, and here is the second. And one chromatid was from the original, and one chromatid is for the daughter cell that will exist later. So here they're chromatids, and eventually these chromosomes will be split, and those will be called, the splitting will make the two chromatids. But right now the chromosomes are aligned at the equator of the cell so that they can be split, and then the chromatids will be pulled to the poles of the cell. But as of right now in metaphase, they sit at the equator. So now we are looking at anaphase, continuing off of metaphase. So again, I outline the cell for you, and I'm going to draw two centrioles at each pole. I will label them for you. Here we go. And now I'm going to draw the spindle fibers. But notice that the spindle fibers are shortening. They are becoming shorter because they are pulling the chromatids towards the poles. And I'm labeling the spindle fibers for you right now. But in metaphase, we had the chromosomes aligned at the equator. But now with anaphase, the chromosomes have been split, and now the chromatids are being pulled towards the poles. So the chromosomes were aligned at the equator in metaphase, but now they have been pulled apart in anaphase, and they are moving towards the two poles.
Alright, so the next phase of mitosis is going to be telophase. A lot of the time telophase is classified as the last stage of mitosis, but we'll cover in a second about why that might not be the case sometimes, and why you might not classify it that way. So during telophase, we have essentially the opposite of prophase. So if you remember prophase, we see the nuclear envelope and the nucleolus disappearing and the chromosomes condensing. In telophase, we see basically the opposite of that. So telophase starts after anaphase, and we can tell the difference between the two of those things when chromosomes have completely stopped moving. So in anaphase, the chromosomes are moving towards each end of the new cells, what will be the new cells. And so once they completely stop moving, then telophase starts. Once they stop moving, we start to see those chromosomes that were tightly condensed start to unravel, and they do that so they can become active and be used for transcription in the new cells that are going to be formed. We also start to see the nuclear envelope starting to reform, and the nucleolus in each new cell beginning to reform as well. And the nucleolus is again where we see the ribosomes starting being replicated and stuff in the new cells. During telophase, we see the mitotic spindle that was pulling those chromosomes towards each side disappear. And during telophase, we see that the cell has two different nuclei. So at this phase, it's what's called binuclear. So the cell has two different nucleus, which you don't see in any of the daughter cells. So this is sort of an intermediate phase. The cell is elongated at this point, so it's sort of long and skinny, and it's started to pinch off a little bit in the middle. And so now, technically, mitosis is done after telophase. That's where a lot of classifications end mitosis. But we have one more step before these cells are completely formed, and that's going to be cytokinesis. Cytokinesis is the process in which these cells start to pinch off from each other and separate. And it's not classified as its own stage of mitosis because it's actually happening during a couple of different stages. So cytokinesis is happening during anaphase. So towards the end of anaphase, you start to see these cells pinching off in the middle a little bit, and that's when the cells start to form a ring around the center that contracts and pinches it off slowly. So cytokinesis is happening during the end of anaphase, and it continues all the way through telophase, and actually past the end of telophase a little bit. And that's what's actually forming these new daughter cells, is once that ring completely contracts, those new daughter cells will have the completely identical DNA and be totally separate from each other. So at that phase, at that point after cytokinesis ends, you have 2 brand new daughter cells and mitosis is finished. And you've successfully replicated your cell.